Welcome to the deep dive. We're here to slice through the complex details and give you the core insights you need. Today, we're jumping into a really exciting space where AI meets biology. We're looking at how these big AI models, kind of like the one behind ChatGPT, are being used to decode the language of our DNA. Yeah, our source material is centered around a new model called JanusDNA. We've got notes, a paper, basically a look at a new blueprint for understanding our genetic code using AI. And our mission really is to unpack what JanusDNA is all about. Why is DNA such a tough nut for AI to crack? And, what are the key innovations here that are pushing things forward? We want you to get the gist fast.
It's a super important area, definitely. I mean, we've seen what large language models can do with text, right? But DNA, it's a whole different ballgame.
How so? It's a sequence, isn't it? Like letters in a sentence.
It is, but the way meaning works is fundamentally different. A single letter in DNA, an A or T or C or G, it doesn't mean much by itself.
Ah, okay.
It's all about the context, where it is, what's around it, and often what's really far away from it in the sequence. That's not how human language usually works.
Right. So standard AI models, they just aren't cut out for that.
They run into some pretty big roadblocks.
Okay, so let's break those down. Why is DNA so tricky for these otherwise really smart AI models? What are the main hurdles?
Well, the first big one we sort of touched on, the lack of inherent semantic meaning. And A, it doesn't carry meaning like the word apple does. Its function comes purely from its position and the sequence around it.
Context is king, basically.
Absolutely. And that leads right into the second major problem, long-range dependencies. In DNA, the elements that control a gene might be thousands, even tens of thousands of base pairs away.
Tens of thousands.
Or even further, the sources mention needing to understand context, potentially up to 1 million base pairs long for some regulatory questions. Now think about AI models trying to pay attention to every single base pair across that distance.
That sounds computationally intense.
Exactly. Traditional attention mechanisms, like in many transformer models, they kind of fall apart. They become incredibly expensive, needing huge amounts of memory and compute power. Getting them to work well beyond maybe 12,000 base pairs is a real challenge.
Okay, so distance is a killer app for standard AI. What else?
Then there's this trade-off between resolution and sequence length. To handle longer sequences, some older methods group nucleotides together. They make words out of them called chimers, like reading ATG as one unit.
Like chunking it up.
Right. It lets you see further, but you lose the fine detail. It reduces resolution, as the paper puts it. And that's a big problem because sometimes a single tiny change, a single letter flip, what we call it SNP, can make all the difference biologically.
So you need to see the big picture and the tiny details.
Precisely. And JanusDNA aims for that single nucleotide resolution. Then there's the need for bidirectional understanding.
Looking both ways.
Yeah. Biological processes in DNA often depend on context from both upstream and downstream before and after a specific point. Think about bidirectional promoters. They can kick off gene activity in both directions along the DNA strand.
Okay.
But many efficient AI training methods, the autoregressive ones, they're built to read in one direction only, like left to right. They miss that looking both ways part.
So the efficient models aren't bidirectional, typically. Are the bidirectional ones inefficient?
That's the fifth point exactly. Approaches like masked language models or MLMs, BERTs is a famous example in text, are bidirectional. They learn by masking out bits of the sequence and predicting them from the surrounding context.
Sounds good.
The catch is low training efficiency. They usually only mask about 15% of the tokens, the DNA letters, in each training step. That means only those 15% are contributing to the learning signal.
Seems wasteful.
It is, relatively speaking. It makes training much slower and less data efficient compared to models where every single token helps the model learn. The paper says it can hinder the model's ability to learn effectively from the entire sequence.
Wow. Okay. So just to recap the challenges. No meaning in single letters, critical connections over huge distances, need for both high resolution and long view. The biology requires looking both ways, and the standard way to look both ways is slow to train. That's quite a list.
It really is. It shows why just taking a standard LLM and throwing DNA at it doesn't quite work out of the box.
Which brings us to JanusDNA. How does it actually try to overcome all of that? This is where the innovations come in, right?
Exactly. JanusDNA brings a few key ideas to the table. The notes highlight two main strategies, a new training method called Janus modeling and a special hybrid architecture. Plus, there's a specific way it handles the DNA's reverse complement.
Okay, let's tackle that first one. Janus modeling, you said it tackles that efficiency versus bidirectionality problem. How does this novel pre-training paradigm work?
Right. It's designed to get you the best of both worlds. You want the training efficiency of those autoregressive models, where every token prediction helps the model learn.
But you need the bidirectional view of the MLM models, looking left and right.
Precisely. Janus model's core idea is this: predict every token using its complete bidirectional context.
Every single token.
Every single one. For any given nucleotide in the sequence, the model's job is to predict that nucleotide using all the information before it and all the information after it.
So every position provides a learning signal.
Exactly. That makes it efficient, like autoregressive models. But because it's using context from both sides for each prediction, it's inherently bidirectional.
Okay, clever. How does it technically do that without, cheating and letting the model see the answer it's trying to predict?
Good question. It uses a two-stage process. First is independent context encoding. The input DNA sequence goes through two separate parallel encoders. One reads left to right, building up context from the past.
The forward pass.
Right. And the other reads right to left, building context from the future relative to any given point.
The backward pass.
Yep. And critically, these two passes are kept completely independent at this stage. The paper stresses this is to ensure no premature information leakage. The forward pass doesn't know what the backward pass knows yet, and vice versa.
Okay, so two independent views, then what?
Then comes stage 2, bidirectional globe fusion. The information, the representations learned by those two independent passes are brought together. They use an attention mechanism here, specifically something called flex attention, which is optimized for efficiency on long sequences to combine the forward and backward views.
So attention stitches the two views together. But how does it avoid that cheating problem during prediction?
That's handled by a carefully designed attention mask during this fusion step. Think of the mask as controlling which parts of the combined information the model is allowed to see when it's trying to predict the nucleotide at, say, position 100.
Okay.
The mask ensures it can see the context from positions 1 to 99 from the forward pass and positions 101 onwards from the backward pass, but it blocks access to the actual nucleotide at position 100 itself.
So it uses everything around the target, not the target itself.
Exactly. Figure 1 in the notes shows this visually. It creates this bidirectional window around the prediction target. That's how it gets bidirectional context for every token without simply copying the answer.
That's a really elegant solution. Like having two independent scouts report back and then using a special lens to combine their reports, focusing only on the surroundings of the point of interest. And the sources say this actually speeds up training compared to MLMs.
It does. The empirical results backed it up. Figure 4 shows models trained with Janus modeling learn faster. They consistently achieve higher accuracy than standard masked modeling for the same amount of training. It really demonstrates superior training efficiency while maintaining robust bidirectional understanding.
Okay, so Janus modeling is the smart training strategy. But what about the model itself, the architecture that has to crunch through potentially a million base pairs? How is that constructed?
Right, that's the second big innovation, the powerful hybrid architecture. JanusDNA doesn't just rely on one type of building block, like only transformers. It cleverly mixes and matches components. Mamba layers, attention, and mixture of experts, or MO.
A hybrid. Why that specific mix?
Each part plays a specific role tailored to the challenges of DNA. The main workhorse for encoding the long sequence is Mamba. Mamba layers are a type of state space model. The key thing is they're known to be much more memory and computationally efficient than standard transformers, especially when dealing with really long DNA sequences.
So Wamba handles the length efficiently.
Yes, it's great for capturing those local contextual dependencies along the sequence without breaking the computational bank. Then they incorporate mixture of experts, MOE layers.
MOE, that sounds familiar. Is that like in some big language models?
Exactly the same principle. Instead of having one giant feed-forward network after the Mamba layers, which can be computationally heavy, MOE uses many smaller, specialized expert networks.
Okay.
When an input comes through, it's routed to only a few of these experts, not all of them. This means you can have a model with a huge total number of parameters, giving it lots of capacity, but the actual computation needed for any single input is much lower because only a fraction of the model is active.
So more power, but not proportionally more cost.
That's the idea. It significantly increases model capacity without proportionally increasing computational costs. The paper mentions they use an auxiliary loss to encourage balanced utilization, basically making sure how the experts get used over time and contribute.
Clever. So Mamba for efficient encoding, MOE for scalable capacity. Where does attention fit back in?
Attention, specifically that efficient flex attention, is used primarily in that bidirectional global fusion step we talked about with Janus modeling. While Mamba efficiently processes the forward and backward sequences independently, you still need a powerful mechanism to integrate those two streams globally.
Ah, so attention connects the Mamba MOE outputs.
Precisely. It allows the model to achieve that global, high-resolution context awareness, linking information across the entire sequence, even the very long ones. And flex attention is chosen because it's optimized for the kind of sparse connections needed here and helps manage memory use.
So it's the combination Mamba for the long haul, MOE for smart scaling, and attention for the global integration.
Exactly. And this specific hybrid design is what enables that key technical capability mentioned repeatedly, processing up to 1 million base pairs at single nucleotide resolution on a single 80 GBB GPU.
A million bases, single nucleotide detail on one GPU. That really underscores the efficiency gains, doesn't it? Makes it accessible.
It's a major step forward in practicality for this kind of large-scale genomic modeling.
Okay, there's one more biological detail we need to cover, right? The fact that DNA is double-stranded, the whole ATCG pairing thing, and the strands run in opposite directions. How does JanusDNA deal with the reverse complement?
Right, that's the third piece, the reverse complement strategy biology reminder. DNA is a double helix. The two strands are complementary, A pairs with T, C with G, and they run in opposite directions. They're anti-parallel.
Okay.
This means a meaningful sequence on one strand, like GATA, has a complementary sequence CTAP on the other strand. But because the other strand runs the opposite way, if you read it biologically, it's TATC.
So GATA on one strand corresponds to TATC on the other, reading in the same direction.
Effectively, yes. And biologically, GATA and TATC might perform the same function, but to a naive AI model, GATA and TATC look like completely different patterns. The model needs to learn their equivalent without having to learn both as separate, unrelated things.
How does JanusDNA solve that?
It's quite direct. As shown in Figure 1b of the source notes, it takes the input DNA sequence, calculates its reverse complement sequence, and then processes both the forward and reverse complement strands independently and in parallel using the identical model.
So it runs the model twice, once on the original, once on the reverse complement.
Essentially, yes, in parallel. Then the learned representations, the outputs from processing both strands, are combined, pooled is the term used, into a single unified enriched representation for that piece of DNA.
I see. So it explicitly considers both strands' perspective.
Exactly. This ensures the model gets a more comprehensive understanding of the biological context. It helps it capture intricate patterns regardless of strand orientation, making it more robust to how information is actually encoded in the double helix.
Okay, so the picture is really coming together now. We have Janus modeling for efficient bidirectional learning, the hybrid Mamba-Mo attention architecture for handling incredible length at high resolution, and this reverse complement strategy to capture the full biological information. That's the how. Did it actually pay off? How did it perform on real tasks?
That's the crucial question, isn't it? The validation. They tested JanusDNA on 3 diverse genomic benchmarks designed to probe different capabilities, especially that global understanding and long-range representation.
All right, let's hear the results.
First, on a standard genomic benchmark set, it performed well, achieved state-of-the-art performance on 3 out of 8 tasks, and was very close on the others. The paper does note this benchmark is quite saturated, meaning it's hard to make big leaps there.
Okay, solid start. What else?
Then they looked at the nucleotide transformer tasks, a broader set of regulatory prediction challenges. Here, the results were really strong. State-of-the-art performance on 12 out of 18 tasks.
12 out of 18. That's impressive.
It is. And here's a really key finding connecting back to the architecture. JanusDNA achieved this while surpassing previous models, including those with 250 times more activated parameters.
Whoa, 250 times?
Yeah, they give the example of beating Informer, which has around 252 million parameters, while JanusDNA, thanks to Mo, might only be activating less than 2 million parameters at inference time.
That's a massive efficiency win. It's not just better, it's doing it much smarter.
Precisely. That Mo layer really pays off. But perhaps the most telling results came from the DNA long bench EQTL tasks. These are specifically designed to test understanding of very long-range interaction sequences, up to 450,000 base pairs, long linking genetic variants to gene expression levels.
The ultimate test for its long-range claims.
Exactly. And on these tasks, JanusDNA got the best performance on 8 out of 9 datasets.
Okay, that really seals the deal on the long-range capability.
It does. The paper highlights that it significantly outperformed the expert model Informer and Caduceus PH, despite using fewer computational parameters. This directly shows its exceptional capability in processing and understanding ultra-long genomic interactions. It proves the design philosophy worked.
That's fantastic. It really shows these innovations aren't just theoretically neat, they deliver tangible results, especially on the hardest problems that set out to solve long-range understanding with efficiency.
Absolutely. The combination of performance and efficiency is the key takeaway from the benchmarks.
So, given this success, what's next? What are the limitations they acknowledge, and where does this research go from here?
Yeah, the paper is quite upfront about limitations and future work. It's a big step, but there's more to do.
Like what?
Well, first, the training data is currently limited. It was mainly trained on the standard human reference genome, HG38. To really understand human variation, future work needs to include diverse human genomes, like from the 1000 Genomes Project, and also expand to non-human species for comparative studies.
Makes sense. More data, broader applicability.
Second, it currently lacks multimodal integration. Right now, it's just looking at the DNA sequence, the A's, T's, C's, and G's.
But biology is messier than that.
Exactly, how the DNA is packaged, chemical modifications on it, epigenetics, and which genes are actually turned on in different cells, transcriptomics, that's all crucial context, especially for understanding cell type specific regulation. Integrating these other data types is a major next step.
Building a more complete picture.
Right. And third, and this is critical, there's a need for experimental and clinical validation. The computational results are strong, but you need to show it works in the lab and potentially in the clinic.
Wet lab validation, not just computational.
Yes. Things like using CRISPR gene editing to test the model's predictions about sequence function or using organoid models, and eventually building clinical collaborations to see if it can help with personalized diagnostics and drug discovery.
Bringing it closer to real-world impact.
That's the goal. The paper wraps up with this vision of JanusDNA evolving into a unified framework linking genome structure, epigenetic regulation, and disease mechanisms, moving beyond just reading the code to modeling the whole system.
Okay, so wrapping this all up for you listening, we've dug into JanusDNA and seen how it uses some really clever tricks, the Janus modeling for efficient, two-way learning, the hybrid Mamba-MOE attention architecture built for scale and detail, and explicitly handling the reverse complement strand. These innovations allow it to tackle some of the toughest problems in using AI for genomics. Understanding connections across vast DNA distances, doing it efficiently, and keeping the resolution fine enough to see critical details.
And that capability, that efficient processing of ultra-long, bidirectional context, it could really change things. How we map genetic variations to function, how we understand those complex regulatory networks spread out across our genome, maybe even how we find new ways to tackle diseases by understanding their genetic roots better.
So think about that future direction mentioned, pulling in epigenetic data, testing it experimentally, moving towards the clinic. If models like JanusDNA really can bridge that gap from the raw DNA sequence through all the layers of regulation, right, to disease, how might that fundamentally change our perspective on health, on disease, on what makes us tick at the most basic biological level? What new questions does that open up for you? That brings us to the end of this deep dive on JanusDNA. Thanks for joining us as we explore these exciting developments at the frontier of AI and genomics. Keep asking questions and keep exploring.